Come on, little fella. Don't be shy. That's it. Come on. These little fellows have been fed diets containing genetically modified potatoes, and all these experiments are done in comparison with the non-genetically modified potatoes, so that you can pick up any differences. We only investigated these three things: growth, development, and immunity, and they were not supposed to have been touched.
But it's that pace of change that's worrying other scientists as well. They say GM food is being rushed onto the market and into our diets without adequate safety tests, and that we're part of one huge experiment.
We got almost 60,000 pages of internal documents. And in these internal documents, it was very clear that the scientists inside the agency said, we need to test these foods for toxicity, allergenicity, immune response, lower nutrition, and for environmental contamination. Page after page of study they said we need. None of this was done.
Today, around a third of the soya from the US is genetically altered. And when it gets to the factory, it's mixed together with ordinary soya. And you never know which is which.
In precautionary science of genetic engineering, it means one question means one career. You ask one question, you get the answer, and you might or might not be able to publish it, but that's the end of your career. What's, I think, very unique in my case is that I survived.
The attack on scientists is very well structured by the biotech industry. It's systematic, it's worldwide, it's very coordinated. It's part of the way that they do business. So anywhere in the world, at any time, if someone finds a problem, they're jumped on. And if the problem is really severe, they get jumped on even more whenever there's something that comes up that can threaten this biotechnology empire.
Companies exist to make money. They don't exist to entirely for the public good. They exist to give profit back to their shareholders.
Missouri. The greatest beneficiary of patented genes was the Monsanto Chemical Company with $4.5 billion profit in 2007. In its report to shareholders, Monsanto predicted these profits would double in the next 5 years to more than $9.5 billion.
Hi, is this Tom? Hi, Tom. This is Jeffrey Smith. You're one of the more than 24 farmers that gave him a call describing problems with pigs that couldn't get pregnant because of the GM corn. Is that right? So what I may do at another time, I'm on my cell phone now, but I might actually, after we talk a bit, I might actually email you and tell you exactly what I've written down about what we've talked about to make sure it's accurate. All right. Thank you very much. Bye-bye. So that was good news. He said that he was willing to talk. His cell phone was dying on the battery. And he said, first thing he said was, is my name going to be in the article you're writing? So I had to put him at ease and tell him no. If not, if he didn't want it to be in the article, it wouldn't be in there. That's why we don't have a lot of whistleblowers. The farmers Farmers are not interested. A lot of scientists are scared about it. And that's the whole state of the art right now, where almost all scientists focused on genetically engineered research are funded directly or indirectly by industry. So we're in a very dangerous situation because there's no real independent science. The biotech industry says all we're doing is adding a gene into the DNA. It's like a Lego where they snap it into place. But that's very untrue. When the single process of insertion can change the natural DNA in many ways that are unpredictable, and they could lead to toxins and allergens and new diseases and antibiotic-resistant diseases and nutritional problems. Most of the soy grown in the United States is Monsanto's Roundup Ready soybeans, where they take the soybeans and they put into the DNA a gene that allows the plant to be sprayed with Monsanto's Roundup herbicide. And it will kill all of the weeds around the plant, but the soybean plant will survive. So 86% of the soybeans in the United States are genetically engineered to withstand Monsanto's herbicide.
Miracle of science or Frankenstein food? How safe is the new ingredient in your diet? Professor Pushtai's lab is at Scotland's Rowett Institute, one of the leading food research centres in Europe. Scientists here are trying to find out whether long-term consumption of GM foods may affect health. The professor is so concerned about the implications of his discovery, he's decided to publicise his findings early.
As a scientist looking at it and actively working on the field, I find that it's very, very unfair to use our fellow citizens as guinea pigs. It was the first experiment because when we started in 1995, there was not a single publication on the potential or the real effects of genetically modified materials with any species, rats, mice, or humans, even though that the humans were already eating it. This is the same variety of potato, Desiree, that we used for genetic engineering in 1995. This is the particular potato variety which is one of the easiest to genetically engineer. And the point of the whole genetic modification experiment was to protect the potato against aphids, which are one of the major pests in Scotland. They attack the potato, the green parts of the potato. And we observed that that particular lectin which we used, which we took from the snowdrop bulbs, did protect the snowdrop bulbs against all sorts of pests attacks. And we had hoped that if we take that particular gene and transfer it, engineer it into the potatoes, that will also be protecting the potato against insect attacks, which in fact it did. After the animals were killed and dissected, we found out that in comparison with the non-genetically modified potatoes, their internal organs developed differently. Some of the internal organs, such as, for example, the gut, increased in size, even though that the rat itself was not growing as fast. Or some of the other tissues, for example, the liver or the kidneys, were not developing as well as the controls. And the conclusions were, and this is important, they found in those data 36 very highly significant differences between the GM-fed animals and the non-GM-fed animals.
Philadelphia, U.S. physicians criticize that after more than 10 years, there are still no long-term studies concerning the consumption of genetic modified food due to the fact that there is no initial experimental data recorded and labeling has not been permitted.
So, even though I'm not a scientist, I worked with more than 30 scientists over 2 years to document the known health risks of genetically engineered foods. And the first part of the book is the documented health risks. And there's 65 different health risks divided into sections. Now, the first one that I chose to highlight within evidence of reactions is Dr. Pustai's work. And the reason why is that Arpad pointed out that it was basically the process itself of genetic engineering that caused this significant damage. So only the genetically engineered potatoes caused the problem. The rats that were fed the natural potatoes along with the same insecticide did not have the same problems. So his brilliant design showed that it wasn't the insecticide that caused the problems. It was somehow the process of creating the genetically engineered potato. And he used the same process that's used to create the other genetically engineered crops on the market.
The immune system takes about 10 days to get in top gear. So if we do a short-term trial, we wouldn't have seen the end result. If I had the choice, I would certainly not eat it till I see at least comparable experimental evidence which we are producing for our genetically modified potatoes. Those 150 seconds were boom, 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 and everybody got the message. Everybody got the message. They asked me, would you eat it? I said, no, certainly not the GM potatoes we worked on. If I could avoid it, I would certainly avoid it. The other piece of the message was that, so what do we want? And I said that what we want is to be more testing, more biological testing, and we should not use, and that they got that. This message originated from us, that they should not use our fellow citizens as human guinea pigs.
When he was asked to speak on television, he had enormous pressure. He was one of the few people on earth that realized that the GM crops on the market might be causing all that damage in the gut and in the brain and in the organs of the entire population. And so with permission from his director, he was interviewed, and then he was a hero for about 2 days at the Rowett Institute. The director praised his work, took over all the press work, put out his own press release, praising it as world-class research. And then the 2 phone calls allegedly placed from the UK Prime Minister's office forwarded through the receptionist to the director. The next morning, Arpad Pustai was fired after 35 years and silenced with threats of a lawsuit.
It is only when we think there was political pressure coming from the top that the situation changed. And then the director, to save his own skin, decided that the best way to deal with the situation, A, to destroy me, B, to make me shut up, So they invoked a contract that whatever they did say on TV, radio, and wrote in the newspapers, I could not deny it. I could not correct it. I couldn't say what was the real situation. And then we looked at the submissions of applications by the biotech companies for those products which we're already eating. And we found out that they were flimsy, they were not scientifically well-founded, and certainly the work which was reported in them did not compare well at all with our very extensive studies.
He said it was a turning point in my life. He said, Jeffrey, I realized what I was doing and what they were doing was diametrically opposed. I was doing safety studies. They were doing as little as possible to get their foods on the market as quickly as possible. So the turning point in his life was realizing that there were other scientists in the world who were not taking care of the health of the world, were not treating science properly. You see, he's on the top level of the world. He expects other scientists to do so. So more than being fired from his job, more than these shocking moments in his life, his greatest shock was to discover that there was this whole world of scientists that would basically allow foods on the market for economic purposes.
So what we have here is 60,000 pages of documents, of internal documents of FDA in these boxes here on what happened when they suppressed their science about GMOs. Going through these documents, we've come up with, we've come up with really the central documents that really demonstrate this suppression. It's really quite remarkable. Number of documents here. You see some of the major scientists at FDA saying these GMO crops are very different. So this is early on. This is the very beginning of the regulation of genetically engineered food in the United States. Scientist after scientist says these genetically engineered foods are not equivalent, not substantially equivalent to conventional food. And that, of course, that was the position that the industry wanted. There's nothing different here. They said, oh no, it's very different. Different proteins, viruses, different organisms inside these foods that create unique health risks. Here we see unique toxicity. These foods could be toxic. You know, there's one document in here that I think is especially important that really shows this history probably better than any other. And this had to do with their compliance in looking at the studies. And here's what the FDA scientists, their own scientists said, and I think it's on page 18,777, yeah, right here. And they said, you know what, we need to look at how these crops change how much agrochemicals, how much pesticides we put in here. We need to see how they change water use, how much water they need, all long-term studies that were needed. Down here, we need to see what about loss of nutrients in the food? What about loss of nutrition? None of that was done. What about loss of diversity in seeds? Long-term studies recommended by the scientists, never done. Here, look at this. Changes in toxin or chemicals in the foods. Study recommended, long-term study, never done. So the scientists asked for these studies, said, let us do them. We think there's a problem here. We've taken a preliminary look. We think there's a problem. But the politicians at the FDA and in the administration at that time said, no, they suppressed the science. And these questions, these studies have never been done. Never been done.
The Flavor Saver tomato was the first genetically engineered food to reach the public. Calgene, the corporation that produced the tomato, had done 3 voluntary feeding studies on rats and found lesions in some of the rats' stomachs.
The question that you're asking is about the Flavor Saver tomato and what went into that. Curiously enough, I was actually one of the outside consultants that was in the last meeting on the Flavor Saver tomato, which wasn't terribly successful commercially. But The FDA scientist went through a long presentation about all the analyses they had done, and her concluding slide was, the flavor saver tomato is a tomato. So they had not found any substantial differences except for the addition of the one extra gene that actually decreased the rate at which they went soft, so they extended the shelf life. And indeed, that was the conclusion, so they had no reason to regulate it on the basis of safety.
No one gets up in the morning saying, I want to go buy a genetically engineered food. They offer no benefits, no more nutrition, no more flavor, no nothing. They only offer risks. That's all they offer to the consumer. And so the average person, of course, rational person would say, why would I buy a food that offers me no new benefits, but only risks? So it was critical for the industry to get these foods out without anyone knowing, because if they knew, they would obviously choose not to buy them.
I don't know how many years of testing you would feel comfortable with, but if I told you it was 25 years, would you feel comfortable? If I told you it was 12 years, would you feel comfortable? 6 years? At what point, how many years of testing and how many different kinds of tests would make you feel comfortable? I will tell you, as I've said before, that because these foods are tested more than any other new kinds of foods that have come on the market, I am more confident about these than I am about picking up a new package of something that the ingredients of which I don't know.
I had a long-term relationship with a group of indigenous communities. And with them, we developed the idea that transgenic materials would come to Mexico. And the problem was that when they come, nobody was there to tell them. So that's when we started thinking we should really prepare a laboratory with capacity to detect the presence of transgenic organisms. And that's where I was helping them to set up that laboratory. And the person who came to train them was my student, David Quist. We were just training, and to train you would have to have a positive control and a negative control. For the positive control, he brought corn from the US, and from the negative control, we thought we should just use the local corn, which of course is going to be clean and wonderful. And the surprise came when the negative controls started coming out positive. That means we started finding transgenic materials where they were not supposed to be. The reason why our findings were so astounding was because it was thought that there was no transgenic corn being planted in Mexico at all. And people wanted it that way. The government, the local communities, the people who lived in the city, they all wanted to have it that way, that there would be no transgenics in Mexico. Why? Because Mexico is the center of origin of corn, and the Mexican government was worried about maintaining the integrity of the land races, the national germplasm. So it was illegal to plant transgenic corn. It is still illegal today.
maize germplasm bank. It's essentially a very, very large seed library. And as you see, the collections are arranged like libraries on a shelf. There are 24,000 samples, unique samples of maize seed. Most of this is land races. These are varieties that were developed by farmers over thousands of years through their selection, and they're quite diverse. These collections are also a source of useful traits for breeders who are looking for things like insect resistance, drought tolerance, grain quality, all kinds of other characteristics. And so sometimes these old land races can be sources of genes for those kinds of characteristics. These things are slowly disappearing from the countryside as farmers adopt newer, improved varieties, and also as farmers leave the countryside, because now, as you know, there's a big problem of rural migration and people from Africa trying to get into Europe, people from Latin America going to cities or going to the United States looking for work. So as these farmers leave the countryside, the varieties that they grow are slowly disappearing.
We're here in central Mexico, in a place where traditionally for many thousands of years corn has been planted in these lands. These are very rich volcanic soils and people plant corn here for many generations. Over the years, corn has changed, and more recently it has been changing because of the introgression of industrial varieties of corn, and most importantly because of the introgression of transgenic corn. We know that there is transgenic corn here, we don't know how much. And we want to be able to tell over the years, over time, whether there is more or less, what kind of transgenic materials are here. Because the transgenic manipulation is invisible, people around here don't even know that they are receiving the transgenics, and they are growing them, and they're passing them on to their children and to their neighbors and so on. So what we're trying to develop is the methods so that they can know what's happening and make their own choices and the decisions about what to do about it. One way to do it, the way most people have done it so far, is to go down to the fields and sample each one of the fields. You take a piece of a leaf and then take it to the lab. But that's very time-consuming and very difficult to do. The possibility that we are developing is the possibility of sampling the air that contains pollen grains from the corn, so that we are, in effect, looking at many fields at the same time, looking at the particles that come in the air, and then detecting the particles of corn, and within those, detecting those pollen grains that are transgenic. It's a challenging, long road that we are following. The next step for us would be to be able to have a piece of machinery that we are developing that we could move around with us. But really our goal in the end is to get ourselves out of the picture, to develop an instrument that we can make available to those people down there, and we want to enable them so that they can go into the internet and through the internet build up a map so that they can see each other on the landscape and in relationship with each other.